Okay, is it recording now? I think
Yeah.
so.
Yeah, it is.
Awesome. So everyone, welcome to PsPablo for week 3. I think we're still mainly on metabolism plus some genetics, and hopefully we can bring some of these really big concepts together, and hopefully also with metabolism, even though it's very confusing, you're slowly finding your kind of big picture looking at metabolism of all these things. And I also would really, what I found helps so much, especially with something like metabolism or something you don't really know, if you watch a PSP or just even go through someone's PSP slides, you can then have a kind of base understanding. So then when you come into the lecture or the tute, it makes more sense. And then it's not as you don't feel as lost or, fully WTF. But yeah, let's get started.
Okay, so you will be doing this week genetics and population genetics, which is, Melonie's pea garden. So the first big thing that is kind of it's kind of important to know is a SNP. So basically a SNP is the common differences between human genomes. So basically what it is a DNA sequence variation occurring with a single nucleotide. So if you look at the picture here, you've got GGC becomes GAC. That's the SNP because that's the difference between one nucleotide in the genome. These are just some information about them. So, 2 random people on the planet anywhere. So me and Ibby are going to differ by 1 base per 100,000. And to be considered a SNP, it has to occur in at least 1% of the population. So about 50% of SNPs are in non-coding regions. So if it's in a non-coding region, that means it's not being expressed by the DNA. But this can also, it's thought to participate with things like promoter activity. So how the promoters work on the DNA, and then stability of the genome and stuff. And yeah, so 25% cause missense mutations, which is, you should know what a missense mutation is already. And then 25% are silent, they have no effect. Cool. And I remember, SNP scissors, because it snips and it snips one out and it puts another one in. That's kind of how I remember it. So the next thing is pharmacogenetics, which is basically this is kind of a new thing, and it's could be a thing in the future that we have to deal with. But basically it's prediction of how someone is going to respond to a drug based on their genome. So you take the person's genomic sample, and then you'd be right, here's all these drugs, this one's going to work best for you. It's thought that the combination of SNPs will determine the drug response, and so already more than 900,000 SNPs can be identified. But yeah, it's not yet widely available. But yeah, cool. Definitions. If you did bio, fantastic, because this will be so much easier for you. If you didn't do bio, that's okay. Try and, you don't have to word for word learn these, but as long as you know what each of these words mean, it will make your life a whole lot easier. Yeah, just we'll put the slides up later, and it's just easy just to know what these are, put them in flashcards, whatever works for you. Yeah, cool. So this next one is the Hardy-Weinberg law, which is probably it's important to know and understand for population genetics and working out how things change and stuff. I think we also touched it, we did touch on it in bio as well. So bio, fantastic. Basically, it's the principle that large randomly mating populations allele frequencies do not change over time. So if you look back to the definitions on the previous slide, we've got an allele as the alternative version of the same gene. So the allele frequency is the frequency of these alleles. So we're thinking of things like eye color, blood type. And this is the equation. So p squared plus 2qpq plus q squared equals 1. You'll need to know this. So screenshot it, write in your notes, whatever. You will need to know that. So basically what this is allowing us to do is it allowing us to determine whether there are influences on a genetic population. And I'm going to go into a few of those in the next slide. So basically, if you've got a large randomly mating population is not in the equilibrium, there may be migration, mutation, or selection. So say you've got like a really large group of meerkats at the zoo. They're going to be mating randomly because there's so many of them, and they're not like saying the zookeepers aren't choosing who the meerkats are mating with. They're just letting them mate randomly. So because it's a large population, but it's sealed off. So they're going to be in equilibrium because they're not having anyone coming in, they're not having anyone coming out, and they're not having anything, they are not having like a disease come in which only affects a few of them. So these meerkats, they're going to be in equilibrium. And so if we, sorry, where was I going with this? So yeah, so basically using this equation, the frequency of the genotypes in the meerkats, say like, I don't know if meerkats have different eye colors, but say they do. The genotype frequencies in the meerkat's eye colors can be predicted from the allele frequencies. So say you've got big A for brown eyes and little a for blue eyes. We can put these in the equation, and if that doesn't equal 1, then we know that there's something else affecting it. But it should equal 1 because they're in a large randomly mating population. And these are all the reasons why this can differ. So we've got things like mutation. So these only result in minor deviations. Say like one of the meerkats mutates to have blue eyes. Migration. So this is where like a group of meerkats from the Sydney Zoo come and join the ones at the Melbourne Zoo, and then all of these ones have got different alleles and they add them to the population. So positive or negative selection. So say if there's a disease that comes in and it selects out one of the genes that the meerkats has, then that will affect the Hardy-Weinberg law. So genetic drift. Sorry, Ali, I'll just finish this and then I'll go back to your question. Genetic drift. So like, I don't know if you did it in biology, but like bottlenecks and founders effects. So it's like, I don't even know. Bottlenecks is like where like you kill off like nearly everyone and there's only like a few left, and then the population grows from there. Or founders effect is where like you take a small group of them and put them on an island, and then the island group starts to grow, but because they've all got similar genes, because they all came from the same small group, then that's what it is. And the other one is non-random mating. So inbreeding or outbreeding. So take a chance if the zookeepers are choosing who mates with who rather than the meerkats just doing it themselves. Yeah. So sorry, did you answer that question, Ivy? Yeah. So is it referring to the proportion of the, yes, that's exactly correct. So if you go back up to, sorry, the slide on Hardy-Weinberg, yeah, this one. So basically, like you could put big A for P and little a for Q, and it would be referring to, so there will be like it would be a percentage, so or a decimal when you do the equation, and that is referring to the population with said allele.
And how that equation is actually like formulated is through that like Punnett square thing.
Yeah.
So that's how you can also remember it.
Yeah. Cool. Okay.
All right, I'm up next, and we're doing lipid metabolism and storage. Yeah, it's not all about bad fats. We'll find out there are good fats too. So pretty much just in general, lipids are used as our sort of like a long-term source of fuel because they just have a higher capacity to oxidize because there's just that many more carbons. And so pretty much they're our like sort of main staple sort of energy for like a long sort of term thing. That being said, that like actually getting energy out of them takes a longer time. And so they're more like a long-term source that's delivered more slowly. So that's pretty much just all we need to know just in general. So we're going to go through digestion, the metabolism, like all the different steps, transport of lipids, because there's a lot of different things that lipids have to go through to get to where we need them. So just digestion, it's not too high yield, so like don't really focus on it too much, but pretty much I've sort of highlighted the things that we really need to focus on. Bile salts, pretty much are these things that our body produces that emulsify the fats into droplets that we call micelles. And this happens in the hollow space of the intestines, like the actual canal or all the food's going through. And then we've got lipases called pancreatic lipases, and these hydrolyze those fats within the micelles to produce fatty acids and monoglycerides. So you know how we usually have triglycerides? So it's a glycerol and then 3 fatty acids. Well, we don't get rid of all the fatty acids. You still have 1 glycerol, 1 fatty acid, and then 2 free fatty acids. That's pretty much how it sort of works. Then next step is that the fatty acids and the monoglycerides enter the actual first line of cells in the intestines. We call them the epitheliums, like the outer layer, and they just diffuse through because they're lipid soluble. And then they get resynthesized in the smooth endoplasmic reticulum into triglycerides. And then the triglycerides, they're packaged with cholesterol and other sort of stuff. And they get coated by phospholipids and proteins. And we call that structure a chylomicron. And we're going to actually just talk about that in the next slide. But chylomicrons are super, super important. And pretty much chylomicrons, these enclosed sort of like lipid fat thingies, they go through exocytosis, they leave the intestine cells, and they go through the lymphatic system, through the lacteals to the rest of the body. Really nice diagram that shows all of this, just sort of to help visualize. Now we'll get to these things called lipoproteins. All lipoproteins are a way for us to transport fats in the blood because they're obviously not, the lipids don't really like water. So obviously it's going to be difficult for us to, get them through the bloodstream, which is all just fluid. So pretty much what we do, well, the body does, sorry, is we get lipids and cholesterol. So usually triglycerides. So we call them triaglycerols, and we'll talk about the difference a bit later. But we chuck them in the middle, and we call it the non-polar core because they're going to be stuff that doesn't like water, and they're on the inside. And then we coat it with phospholipids, which we know have those hydrophilic heads, and other proteins we call apolipoproteins. And pretty much they're the protein component, and they help sort of with all the receptor mediated sort of stuff, and endocytosis, exocytosis. And there are 4 classes of these lipoproteins. We have those chylomicrons we talked about before. They're the least dense, they're the biggest ones. And then we go to smaller ones. Then we have the VLDLs, very low density lipoproteins, and then high density lipoproteins. We'll find out later the high density lipoproteins are our friends. They're the good ones. So how transport sort of works and where these sort of lipoproteins come into play is when we're trying to get our cholesterols and triglycerols around the body to where they're sort of needed. So we start off with them being in the lymph, in these chylomicrons. Then we have these lipases, and these lipases just attacking things. They're really just aggressive, I guess. And so pretty much they attack the chylomicrons. They free the fatty acids inside, and that's only when we're going to start using them. So where they're needed, sort of thing. The remainder of chylomicrons go through a couple of different things. So they usually get taken to the liver, where we sort of add them to what we call the body's cholesterol pool, which is just the store of cholesterol that we have. So when we need it, we just mobilize them. Or alternatively, they're processed into VLDLs. So there's that second sort of density layer. And they are that's the form we use to transport across the body after the initial sort of being taken up into the lymph. So when we want to get through the bloodstream, we use VLDLs. And then again, lipase, gotta love lipase. It attacks the VLDLs and it frees those fatty acids again, but it also forms LDLs, which is now the next density. So it pretty much frees a bit of the contents to make it even denser. And LDLs are really rich in cholesterol. That's why we sort of, the body does this sort of process, because if we only want to use cholesterol and only need the cholesterol, not the fatty acids, we'll use the LDL form to, get it to places. And then again, we're going to have different sort of things that are going to happen. The LDLs go back to the liver, and they can also make up that cholesterol pool for the body. Finally, we have HDLs, and they're the good ones that we talked about before. Pretty much what they're going to do is if there's excess cholesterol in the body that we don't need, because cholesterol, we find out, is not great if we have too much of it. What it does is it collects it, takes it to liver, which then takes it to kidneys, and we get rid of it through urine. So pretty much it's that good cholesterol, the one that stops us from having too much. Again, beautiful diagram to help show this. It's quite a complicated process. I guess just read the slide a couple of times, look at the diagram, visualize, and just see it's like a process, like a journey of our lipids to somewhere. And yeah, that's a great meme, I thought, because lipases are aggressive as hell. So all right, let's go. Well, we just talked about how we sort of need cholesterol in our tissues, and LDLs are what get us that cholesterol to the places we need. But how does it actually get inside? Because they're relatively big. They're not like something that's quite small that can just diffuse across. So what we usually do is there's a process called receptor-mediated endocytosis. And that's what we talked about, those apolipoproteins before. They're like these protein receptors on the outside of the lipoprotein, and they can connect with the cell's receptors and then enter through endocytosis. And what's cool about the body is we need to be efficient, right? We can't just keep making these receptors once they're used up, and when they enter like the cell with the, you know how like when endocytosis, the receptor gets taken up as well because you get that vesicle forming. So we need a way to recycle it. And so we have the Raphia and the Golgi apparatus that help with that. Like they make the receptors, but then they also help with that recycling process. Yeah, so that's we just said before, LDLs are taken up with the endosome, which fuses. And then we have the lysosomes, these organelles in our bodies that pretty much have just all these enzymes that destroy stuff. They fuse with our endosome and they break down sort of the lipoprotein, the LDL, to free up those amino acids, the cholesterol, the fatty acids. Cool. Now we'll get to mobilization. So I'm going to now talk about the difference between triglycerides and triacylglycerols. Pretty much the same thing. Like they're not actually different like concepts. It's the triglycerides a common name for the general structure, whereas triacylglycerols are that one glycerol and the 3 fatty acids. That's like the actual name. So I guess get familiar with both of them, because lecturers, depending on what like branch or like what facility they're from, you know, they're going to start using different terms if they're specific or not specific. So just get used to the fact that they sort of are the same. Yeah, Zach, sure. So pretty much the difference between these two is that triglycerides, that's just a more general term, a common term. And it just means, you know, just in general, like a fatty acids attached to a glycerol, things like that. But doesn't necessarily, like, it's not a more specific term. The body uses triacylglycerols, which is just that one glycerol and 3 fatty acids. So it's just, they're pretty much, you can think of them as interchangeable, but be aware that like the tags pretty much is what you sort of want to be using. So anyway, in terms of mobilization, we first turn these things into the fatty acids because they're the components we sort of want, and we transport them in the blood via serum albumin. So why don't we use all those other things? Well, those other transports, like the lipoproteins, is when we haven't processed these things in the liver yet. They're still like outside, and we're just going to get them to where they're needed. But once you've stored them in the liver and the adipose tissues, when you get them out, they're going to just be fatty acids. They don't have to be attached to the triglyceride and stuff. And that's why we can just use this albumin, which is a protein in the blood that can just help us transport. So that's the difference, I guess, between the two. And so once you reach target cells, they dissociate from the albumin and they just go through. Yeah, that's pretty much all we need to go. Metabolism, the key slide of all of like this section for today. It's that fatty acids go through a process called beta oxidation in the mitochondrial matrix. And what this process is, it's like a cycle sort of thing. And you go through that cycle 7 times. And each time you go through the cycle, 2 carbons are taken off to form acetyl-CoA. So overall, the products you make are 8 acetyl-CoA, which then go to the citric acid cycle, and then 7 FADH2 and 7 NADH, which go to the electron transport chain. So that's why fats can make so much more energy than carbohydrates, which only turn to a limited amount of acetyl-CoA, which then becomes these FADH2s and NADHs. Whereas just by themselves, lipids produce that much. So that's why that's sort of that sort of greater energy source.
Also, can I just add a quick point? It doesn't necessarily go through 7 times. So that also just will depend on the length of like the fatty acid that's being done. But usually it's 7 times. I think that's the one can't remember what that actual lipid is called, but yeah.
Awesome, thanks, Lily. Yeah, fun fact. In terms of fatty acid synthesis, it's when we're actually forming them or like want to make them. So pretty much if there's too much energy in the body, our liver cells just start doing this. And pretty much they get acetyl-CoA, which is a 2-carbon thing, add it with CO2 and ATP to get something called malonyl-CoA, which is a 3-carbon thing. And then we start, doing pretty much the reverse. We start adding 2 carbons every time. And this process requires ATP and NADPH. And then we get a sort of approximately, usually that's what Lee was saying, it's usually 16 carbons, but sometimes it can be more or less. But we get these fatty acids, which then become, like we can make them to either phospholipids or tags, triacylglycerols. Just be aware, we'll touch on this like later in the year, but if there's high levels of carbon, like carbohydrates, sorry, not carbon, carbohydrates, then and there's low levels of fat, then the synthesis level increases. But when we're in that fasted state, so we're not eating a lot, and we have high fats in our diet, we're going to decrease this process. Like it's sort of like a way our body sort of makes sure we're in equilibrium. Yeah. Now we're going to move on to cholesterol synthesis. So there's a lot to take in with this lipid stuff. So just like, I guess, take it with a pinch of salt. Like you're not going to need to know everything. Like Norma's right when she says don't memorize these reaction pathways because you really don't need to. But pretty much cholesterol is something that doesn't actually give us energy, and we can't actually break it down. It's more something that, you know, is in our membranes to help stability, or it's a precursor for steroids, and that's super important. You're going to find out later on in the year how important steroids are in a lot of processes, and pretty much their base is cholesterol. So that's why we really need cholesterol. And unfortunately, we can only get it by direct absorption when we eat it, and our body tries to get rid of it as well through urine. And so we're trying to get that balance, because if you have too much, we'll see later on in the slides, that's a bad thing. Importantly, just like ignore all the steps in the reaction. Something you just need to note is there's an enzyme called HMG-CoA reductase, and that's just a drug we now sort of target. That's an enzyme we target with drugs when we want to lower lipid, cholesterol levels if they're too high. And yeah, I think that's pretty much all you really need to know. Now I'm going to give you a bit of warning. Spoilers ahead for semester 2, but I just a bit of a brief sort of thing. If there is too much cholesterol, we can get a condition happening called atherosclerosis, which is pretty much plaque buildup in the vessels. And that's why, and that can cause things like stroke and things like that. So we don't want that to happen. And that's why it's really important that we regulate our cholesterol levels. I think this might be the final slide, not sure, but ketones are this alternative energy source for our body. And it's usually when, like sometimes in diabetes, for example, we can't make malonyl-CoA. So pretty much it's an alternative source of energy if we require it. And things like the brain love ketones over anything else because they can cross the blood-brain barrier. So that's why ketones are also this important pathway. And they're made by acetyl-CoA. It's pretty much, they get, I think, broken down. Sorry, acetyl-CoA can either go through oxidation, as we said before, like the citric acid cycle, or alternatively, it can be made into ketones, which can be used by other places in the cell. Just be aware that the liver can't actually use it. So pretty much it only makes it to give it to other areas of the body. Yeah, and that's it from me.
Firstly, we just had a question in the chat about why lipids produce more energy than carbohydrates. And I think as the way that I understand it is that, like, if you go through all the steps, you'll see as you're producing stuff, you produce more like NADHs and FADHs and ATPs and all that stuff when you're at the end of the day, produce more ATP when you break down a lipid versus like one molecule of food.
Yeah, and just like adding on to Liza's point, it's just like as many acetyl-CoAs as you can get. That's how I like to think of it. Like you're breaking down that fatty acid into acetyl-CoA, and then every time that fatty acid might make, let's say, break down into like 8 acetyl-CoAs, each one of those go into this citric acid cycle, versus just in like glycolysis from just carbs, only 2 go in. So that's why, as Liza said, like you're going to end up with more.
Yeah, exactly. So we'll do alcohol metabolism. I don't know. It probably is an lecture or a workshop. Like they kind of go over it pretty briefly, but it's just 2 slides essentially of what you need to know. So the parts involved are the important things. So firstly, that it goes ethanol, acetaldehyde, and then to acetate. Just like, so all those things are kind of like toxic. So yeah, literally poison. So that's why it keeps breaking it down, because like this gets prioritized because all these things are like bad for your body. So they need to try and make it into acetyl-CoA. But yeah, then the enzymes, I honestly don't think you remember them, but they're there if you want to. The most important thing with this is that this process of breaking down the ethanol, both steps, so first, the first step to acetaldehyde and then the next step to acetate, produce NADH, which kind of seems, it's a bit counterintuitive, like it seems like that would be good, but what it does is it like kind of puts you out of whack because it uses up your NAD pluses, and it changes the ratio of NAD plus to NADH. And so when you see like all the effects of alcohol or alcohol metabolism, they're almost all related to that. Also, just an important thing to note, which isn't the only thing that's not related to that, is that that how much alcohol you have inside you doesn't impact the rate of metabolism. So most things, if you have more, it'll get metabolized faster. But because the enzyme there's like a limited amount of the enzymes that break down alcohol, they get like, I don't know what the word is, like it's all used up. So essentially, like you can't break down more than, what's the word though?
Saturated.
Yeah, saturated. So you can't break down more than, I think it's like 1 gram per hour. So yeah, important to remember that. You'll go back to like that sort of like that concept in farm, but just remember with alcohol, you're always going to be breaking down the same amount every hour. So yeah, the effects of the NADH buildup or /NADH NAD plus shortage. There's a lot, but if you like, essentially it's all most things in metabolism go NAD plus to NADH, and so instead it all gets like reversed, or slowed down. So for example, a lot of energy pathways like glycolysis, beta oxidation, Krebs cycle all require NAD plus, so they all slow down, causing fatigue because you have less ATP. The anaerobic pathway for the breakdown of pyruvate is favored for the same reasons, so you end up with lactic acidosis. Fatty acids can't undergo beta oxidation, as in the first point. So instead, they undergo tag synthesis, which I guess is what you were just speaking about in the liver. So that's why you end up with fatty liver or cirrhosis, like liver damage. Also, the toxic components cause inflammation of the GIT lining, so which reduces the ability to absorb thiamine, which is something that's useful. Well, not useful; it's needed to convert pyruvate to acetyl-CoA. So again, also that leads to even more fatigue. But yeah, that's about it.
Okay. Wait, is this me image or is it you? Hang on, I'll just check. Okay, it's me. Awesome. Can't even remember the slides I made. So we're going to be looking at chromosome disorders now, and just a little background on it. All kind of stems from the chromosomes, if that makes sense. So it's important to know the different types of chromosomes that we can have. And there's 3 main types, and it's all dependent on where the centromere, which is the little circle in the middle, is located. So in each chromosome, we have our 2 chromatids that are stuck together. That's why there's like the 2 lines. And so your metacentric is where your centromere is in the middle, and it kind of makes sense, like M for middle, that's how I remembered it. Then we have submetacentric, which is where the centromere is like below the middle, or if you can see in the photo, it's kind of leaning more towards one of the ends. And then the last one is acrocentric, and that's probably the most important one to know, only because when you have a centromere at one of the ends, it means that the little arms, they are called satellites in this case, and they don't really have as much DNA, which makes sense because if you think about it, if it's a really small part of a chromosome, it's not going to be able to have as much DNA. So the acrocentric are the ones I would say to just remember the most, because it will become a bit more important later on. Just some more key terms that are important to remember. The short arm is always called the P arm, and that's because it stands, I think it stands for petite, unless they just told us that to remember it. And then the Q arm is the long arm. Just some words that would be helpful to know and recognize. Okay, cool. So I think you might have seen this in bio before if you did it, but we basically have a karyotype, which they do a test on your DNA, and they can make this image where all your chromosomes are in order. and they're put in order from largest to smallest. So as you can see in the image, we start with our metacentric at the top and it goes down to your acrocentric. And then obviously you have your sex chromosomes as well. We have 22 pairs of autosomes and then 1 pair of sex chromosome. And what's important to know, and what you'll start seeing more, is in the bottom left corner, this is how we can describe someone's karyotype. So we write the number, the total number of chromosomes, and then we do a comma, their sex chromosomes, and then comma, a description of any observed abnormality. So obviously when we say normal male, what we're saying is we're just saying like the general karyotype of someone that has their complete chromosomes. So that would just be written as 46, XY, and there's no abnormalities, which means you wouldn't have that third section. If we move down to someone with Down syndrome, we would now have 47 chromosomes because there's an additional one. And if it's a female, it would be XX. And then we have that third part where we say plus 21 because they have an additional 21st chromosome. And then if we look at the last one, if a female has a deletion of the short arm of chromosome 5, this is where all your key terms come in handy, because you would have 46, because they still have 46 chromosomes, but they just don't have a full one, XX, and then you would write del for deletion, and then 5, and then that's where the P for the short arm would come in to use. Okay, so we're going to start by looking at numerical abnormalities. So when we have a difference in the amount of chromosomes. So the first one we're going to look at, and some of this stuff I think you do look at in bio, so that's helpful if you've done it. But the first one is called an aneuploidy, and that's when you just have like either additional chromosomes or a lack of chromosomes. So instead of having 46, you might have 47 or you might have 45. A monozyme, I don't even know how to pronounce it, monozyme is a loss of a single chromosome, but you don't really see this much like later on in med because it's usually lethal before term. So they're not really going to give you like clinical cases of a monozyme because like most fetuses will not make it to birth. Then we have a trisomy, which is the most common, and that's the gain of a single chromosome. And an example of a trisomy is Down syndrome. So that's a lot more common and one you'll see the most. And then a tetrasomy is when you have a gain of 2 chromosomes. Again, I don't think I ever saw that once we learned it. Something that's important to kind of look at is how an aneuploidy can form. So how can someone get this additional chromosome? And there's 3 options, because you can either get it during the first part of meiosis, the second part of meiosis, or in mitosis. So if we start with the first, if we start with this one, as you can see in the top, so what happens is if we start with our normal like fertilized egg, instead of it splitting into halves, what happens is the 2 chromosomes stay together. And so when they split, instead of having 1 in each, we now have 2 like chromosomes in each. And so when it's fertilized, when the sperm comes, we, instead of having 1 + 1 to give us 2, you now have 2 + 1 to give you 3. And as you can see in the bottom line, the other part will have 0. So when you add 1, that's how you could get to a monozyme, because then you're missing one of the chromosomes. They don't really ask questions about this. It's just like nice to be able to conceptualize. So then if we look at meiosis 2, we've still got our chromosomes which have split into each. But now, if you remember in meiosis, the chromosomes split into like 2 separate bits again. And what can happen is if we look at the top line, in this case, they don't separate into those 2 bits, and they both go into 1 cell, which means the same. You would now get 2 joining with the 1 from the sperm, which would give you the 3 chromosomes, and the other one, which had 0, would give you the 1 monozyme. And then the third one, which is probably the most important because it's like more clinically relevant, if we look at aneuploidy because of mitosis. Basically, once you've already got your egg, like your fertilized egg, your zygote, I'm pretty sure it's called, what can happen is if it starts splitting incorrectly, some of your cells, so this, what the key part is, that some of your cells can still be splitting normally. So because like mitosis, each cell is splitting on its own and independent of the others, what can happen is some of the cells in your body will start to split abnormally, and you could get like a trisomy, for example. However, some can still split normally, and that can form what's called mosaicism. It's a type of, like, some people with Down syndrome have it, and it means that some cells in their body have a normal amount of 46 chromosomes, and other cells in their body have 47 chromosomes, because only some of their cells had this non-disjunction occur. So the three most important aneuploidies to remember, and they do love asking questions on them, so definitely remember them. We have Edwards, Patau, and Downs. So Downs, I feel like we learn about at school. Like, for me, it was the easiest one to remember. And the most important thing to remember is that it's trisomy 21. So in each case, you want to remember the chromosome that there's, like, which chromosome has an additional one. You don't have to worry about the facts about like 1 in 700 live birth. They don't really ask about that. What's important to see is you can see that it says that 95% are a complete trisomy, which means that it formed during meiosis. And then 1% is mosaicism. So that's the cases where it happens during mitosis. And then we'll have a look at chromosome rearrangement in a little bit. Then, back to the other one quickly, just two more things to look at is Edwards is trisomy 18 and Patau is trisomy 13. And those are really important. Just try think of a way to remember which one's Edwards and which one's Patau, because a really common question I remember seeing a lot is like, what, like, trisomy of which chromosome form like leads to Edwards syndrome. So then you need to be able to remember that, okay, it's trisomy 18.
I just remember that by Patau. It's like smaller, like there's less words, letters in it, and it's a smaller number.
Smart, yeah. Then we have sex chromosome aneuploidy. So that's when we have an abnormal number of X or Y chromosomes. And again, the two most important to remember are Klinefelter and Turner. So if we start with Klinefelter, that's XXY. And the most important thing to remember is that as soon as there is a Y chromosome, the person is automatically classified as like a male, if that makes sense. So even though there's an additional X, this person is still a male. However, if you think about it, they've got this additional genetic information. And so their like most prominent physical characteristics are things like breast enlargement and small testes. So they're kind of leaning away from like your normal male characteristics, if that makes sense. And then Turner syndrome is when you just have one X chromosome. So again, because there's no Y, they're considered to be a female. However, they have less genetic information. So you would find, I just put this photo in because it was like a key feature. They have redundant neck skin, and they basically have this really prominent neck. So that's a key feature of Turner syndrome. And also treatment for that would be growth hormone and estrogen replacement, because if you think about it, you're trying to like increase those female characteristics. Then we have polyploidy, which is a lot less common, but that's basically when we now have a multiple of our entire genetic information. So literally you take your karyotype and you multiply it by, for example, 2 or 3. The most common is a multiple of 3, a triploidy, which is where you get, for example, 69XXX or 69XXY. And to be honest, they don't really ask questions about polyploidies, but it can occur because you either have an egg which has been fertilized by 2 sperm, so you get that extra genetic information, or an egg fertilized by a sperm which already has that extra, like it's a diploid sperm, so it has extra genetic information. It's important, well, it was just a fun fact, to be honest, that polyploidy naturally occurs in your bone marrow cells. So your megakaryocytes are cells which grow super, super big, and then they break down to form platelets. And so they actually all have this polyploidy because it allows them to grow bigger, and they also never undergo cell division. So I just thought that was a cool little fact to add. Okay. So we've just looked at the numerical abnormalities, and the second section is when you can have structural abnormalities. So when you have like something that happens, sorry, I dropped my pen and Lily's laughing now. Okay, so structural chromosomal abnormalities is when you have something that goes wrong in your chromosome. So most of them are quite self-explanatory. So if we look at insertion, that's when you take DNA that wasn't in one of your chromosomes and you add it to a chromosome. So if you can see, it's moved from this chromosome and it's made the second one longer. Translocation is when you take part from a different chromosome and add it to a new part. As you can see in translocation, it kind of like swaps, like part A from chromosome 1 will swap with part A from chromosome 2, for example. Deletion is obviously just the opposite of insertion, where you lose genetic information. They're all quite self-explanatory, to be completely honest. Duplication, you double that part of genetic information. And an inversion is basically where it flips. So like part A and part B will just like swap places. Okay, and then you can have, so with our translocations, there's a few different ways that they can occur. So with a reciprocal translocation, I quite like these diagrams. They help me to understand it. It's basically, as you can see, where like the blue and the pink bit will both be cut off and they will swap spots. So it's called reciprocal because they each lose a bit and they each gain a new bit. If we look at insertional, that's where it just leaves one chromosome and enters another. So it's not reciprocal because both chromosomes are not gaining information. And then if we look at the Robertsonian, this is where our acrocentric chromosomes come back into play, because what happens in a Robertsonian is both of our little short tails will break off and the long tails of the two that have just lost the little bit of information will join together. So you go from having 2 chromosomes to now having 1 chromosome, because the little short tails don't have enough genetic information to constitute a chromosome on their own. And you'll learn a bit more about this later when you look at some types of cancer, I think it was. Yeah. So balanced rearrangement is basically when we had those reciprocal translocations, even though the chromosomes have lost part of their genetic information, because usually they just swap with somewhere else. Usually it's called balanced rearrangement. And in most cases, it's usually harmless because you still have all that genetic information. What's important to know though is it can increase your risk of your child having an unbalanced chromosome complement, because basically what happens is, because your chromosomes, like your information is now in a place where it's not meant to be, when your chromosomes pair up, like during meiosis, the genetic information in each cell is like a little bit different, if that makes sense. So when they split, when your 2 chromosomes split now, the child can have more of some genetic information or less, depending on what part of the chromosome they get. Yeah, and I was really bored in ISO last year, and I used to make these little 1-page summaries. So this is a 1-page summary I made of chromosome disorders. So it's basically got everything important you might need. So yeah, that's it.
Awesome. All right, so I'm doing 2 that I don't think you actually have lectures on, but you have tutes on them, and I'm pretty sure that these were the 2. So I'm doing great metabolic rate and the role of the liver. These are actually the 2 most interesting tutes I actually found in Sem 1. So get excited for them. The great metabolic rate is actually probably the most applicable thing from metabolism. And it's just drawing all metabolism kind of together, but mainly just like carbs and lipid like usage. So essentially they're asking, during, let's say, a race of 60 minutes, what is your energy source going to be? Is it going to be the same throughout? Or are you going to like fluctuate? And the answer, obviously, from the diagram is that you're going to fluctuate. So if we have to see the dark line up here, this is the carb usage throughout the time periods and the fat usage. And this is essentially an important diagram to just know, because it just makes everything easier and understanding. But just to like a bit of a, it's not a warning, but just like to note, lots of this stuff is actually very, if you understand like what metabolism is and like the energy sources we get and like where everything's stored, what you're actually going to use your energy for is quite like kind of almost self-explanatory or logical. Yeah, so these are just like some good pointers to go out before we start. And we've pretty much touched on most of them, but first, fatty acids, they're more energy dense, yet they take longer to get the energy out of them, and also they take longer to mobilize. Carbs, they're always available and they're not hard to process. Just some keywords. So if something is classified as dietary, it means that we've essentially almost just finished eating it. So it needs to be broken down ASAP, or otherwise it will be stored. And if something is stored, it has the potential for energy, and it needs to be transported and broken down before it can be used. And also just really important to note is that the body always needs energy. You always have a chemical process going out. And this is kind of why you always need a constant blood sugar or thereabout. And we'll return to this point a bit later on. So if I just go back to this time frame here, this is 60 minutes. And these kind of green dots are kind of the key times to know. And then we have the start here as well. So if we think about a race, and when we talk about metabolic race, we just mean 60-minute period if you were exercising. So when you're resting or just before you're about to race, most of your energy actually comes from your dietary fatty acids. This is just because they're a high energy source. And even though we say that fatty acids take a long time to mobilize because they've come out of your diet, essentially they're available for usage. Then the first sprint that you're going to do to get started on your workout is going to be creatine phosphate in your muscles. Doesn't matter how what this is, just essentially know it's in your muscles, going to be used very quick. It's a quick source of energy, but it's not in abundance. So you're going to quickly run out of this. Then as you're still like, catching your breath, lots of the processes are going to be anaerobic, and you're going to have an anaerobic pathway for your dietary glucose. So this is going to be things like lactic acid production. And this is instead of the glucose entering glycolysis, then citric acid cycle, it just goes straight to make lactic acid, and you get a small amount of energy from that. Then over time, as you're getting your oxygen in, you're kind of adjusting to the race, your muscles begin to mobilize G6P, which goes straight to glycolysis, and glycogen is mobilized by the liver. So here's the start. We start off with dietary fat, metabolize. Then carbs are going to quickly increase as our main source of energy. This is also going to use times we're going to be anaerobic, and here we're going to be aerobic respiration of these carbs. And then at 5-minute marks, the carbs reach the highest percentage of energy gotten from carbs. Also, just I can't actually read the chat, so if someone would, or anyone has any questions, just let me know. I speak them out. Easy. So this is where we're at now.
One question.
Yeah, sorry.
So is the phosphate shown in this graph, or it's just kind of like assumed that it's quick energy at the beginning?
It's assumed as quick energy, so that's why it's not in the graph.
Makes sense.
Yeah, so this is fat. We've got to start with mainly fat. Then we have a point where carbs are really taking off. And then we're at this 5-minute mark now. Okay. So we're beginning to mobilize the glycogen in our liver, and also glucose is used in our muscles and also being mobilized from some small storages in the muscles. Then at the 30-minute mark, there's actually I think that I've seen a couple of questions. This is when you have even usage, or it's not even in the sense of you're using 500 grams of fat, 500 grams of glucose, whatever. It's when both of your energy needs, all of your energy needs are made both out of metabolism of fats and also glucose. So this one, they're even. And this is this 30-minute mark here, okay? Because your fats because even though your stored fats take a while to like kind of churn up and give you the energy, they've slowly been rising. We've slowly started mobilizing, and this is where they've kind of started to meet the main needs. And also here, your carb sources are kind of running out, glycogen sources. And that's kind of where we go towards the end. So we end with mainly fat, mobilization of fats, and we don't have as much like glycogen storages. this is kind of just kind of a fun fact. If you exercise for longer than 60 minutes, usually your fat is burned. And also that's why they say to exercise in the morning, because you don't have as many of these reserves, therefore you're going to burn more calories. You will do more of this later, but this is just like random times during the day. What energy sources are you going to use? All the purple is dietary, which makes sense. You know, this is blood glucose, and this is just times throughout the day. Your blood glucose is mainly going to be maintained by dietary. That's why we eat quite a bit. We probably eat a bit more than we need to, but this is what's keeping our kind of like spiking up our blood glucose. But there's times during the day where we might not eat, but our blood glucose levels still have to be very constant. This is where we would use glyconeogenesis, which is this orange one, which you're mainly going to use overnight. So this occurs after 4 hours and 8 hours of starve. We call this starving, just sleeping really. This is when your proteins are used or like glycogen or glucose sources. Glycogenolysis is when we're kind of breaking down our glycogen stores. So like kind of freeing up those glycogen stores to make some glucose. So this is when, as our glycogen stores are breaking, our body needs to find other places to get the blood glucose from. So that's why we're going to create some. And you would have gone through, maybe we're doing it next week, or you would have done it before, like where these sources come from or breaking down of the glycogen. All right. Now we're on to the role of the liver and just where all these, we get our energy from or where it's used. This is the one that I think was my favorite shoot. It's actually pretty cool because you get like a case, and not to ruin anything, but you kind of work through it and then you kind of find where all the issues might be, and you kind of actually feel like you're being a doctor, even though we still have a long way to go. But as we can see in all these pictures, this is for carbs, fats, and this is proteins. The liver is like a central player in all of these things. You don't need to know these exact kind of pathways at all, but as we can just see, the liver is central to all of these. You will eventually be able to draw most of this. So I actually think the liver is pretty awesome. So I've made kind of like a little fun meme thing, but it's actually very like applicable. So the liver is the main processing station of all like nutrients. So it receives all the nutrients from like that we digest. It then sorts and distributes and responds to the energy needs of the body. So kind of why I wrote like the liver is the ideal girl or guy. So these are like some kind of fun points about it. So it plans for the future. So if we have, we just had a massive carb, we just carb loaded, you know, we're not actually going to be able to eat all of that, use all of that energy. So the liver's like, you know what, I could be greedy and spend all the energy now, but why do that? That would be a waste. What I'm actually going to do is I'm going to store some of this food and I'm going to make it into glycogen. I'm going to store it as fat. And then that means that when we actually, in times of need, maybe as we're sleeping or times where we can't get access to food as fast, we like have a ready sources of energy. So it plans for the future. Another thing, it helps out in the now. So it is constantly distributing glucose to all organs that needs it, and also synthesizing and sending out fatty acids to places that need energy. So it kind of works as like this kind of great middle ground that or honing center that then sends and responds to everything. It's like a central control for kind of the energy needs of your body. I said it's a great listener because it responds directly to hormones. So these are things like insulin, glucagon, things like that. And it doesn't complain, it just follows. So when insulin is telling it, you know, I need the blood glucose is very, very low, I think. It takes up the glucose and it's very high. It takes up glucose and stops the breakdown of glycogen because it says, you know what, we could use our sources, but we're going to save those for later. We're just going to take up the energy we've gotten from our recent meal. And then the reverse for glucagon, it also puts others before itself. What Ibby was saying earlier was that it actually makes ketones, which you make ketones usually when you're starving. So towards probably as you're waking up and people who have diabetes make an excessive amount of ketones. But it itself can't even use the ketones. So it's kind of like it's a great team player. So it sends these ketones to, the brain, and we'll touch on that later with other things. And it itself can't use ketones, which is actually an important point to note. So whilst the liver makes ketones, it can't use them for its own body. Yeah, I'll let you back in. It also, I said, cuts out and deals with the toxic people. So it converts like excess nitrogen to urea, and it's also the side of alcohol metabolism that, deals with lots of the shit that actually we put into our body. And then it also just ensures stability. And I said this stability being cholesterol, because this is one of the sites of cholesterol synthesis, the liver. And the liver and cholesterol is actually used in cell membranes to, keep that strength of the cell membrane. But yeah, the liver's pretty awesome. One super important point is that which I put in yellow, glucose in the liver deals with glucose 6-phosphate, but you can imagine this phosphate group is like polarized. So this glucose with the phosphate, this is the glucose and the phosphate attached, it can't actually cross evenly through the cell membrane, go into the bloodstream to be sent out. So before the liver releases glucose to the body, it has to dephosphorylate it. So you have just the glucose, which can then freely travel into the bloodstream, cross cell membranes, and do all the jazz that it needs to then get to the places it needs to. So that's key. So before glucose can enter the bloodstream, it has to be dephosphorylated. So these are just like other things in the body and kind of what their role is in metabolism, energy sources, essentially giving us energy or storing energy. So we have our adipose tissue. This synthesizes and then stores tags, allowing them to be broken down later. Important to note, it doesn't make the fatty acids, it just receives fatty acids, pairs it with glycerol, and then you have your tags, and it then stores in the adipose tissue, and it then breaks it down when it needs to be used for energy, as Ivy was saying, attaches it to that albumin. Heart muscle, so this uses mainly fatty acids, but it doesn't have much capacity for storage. Our red blood cells undergoes anaerobic oxidation of glucose. The brain, very important, it uses glucose as its primary source of energy, but it has no energy stores, which kind of makes sense, like it's just a mass of nerves. And when it's starving, it will use ketones. And skeletal muscle, it produces all the energy it needs from glucose. It can be some stored, but mainly it receives it from the liver. Takes up amino acids for protein synthesis, yet it can also break down. So that's why if you have like severe starving, the proteins in your muscles are actually being used, and that's used for gluconeogenesis. Yeah, and then this is just a diagram kind of showing all the kind of major points. This is actually from a lecture you will do next week, but it's nice to have just kind of a broad understanding of all what these things do. And also, this is just knowledge that you'll gain also from the Great Metabolic Race and also the role of the liver. You'll gain this from your tutes in this week, which you should be looking forward to. That is it for our PSP for this week. I'm going to stop sharing. Did anyone have any questions? No? Awesome. I'm just reading someone's question. Easy. All right. So I guess we'll see everyone next week. Have a fun time this week. See
Yeah,
ya.
thanks for all coming.
Thank you.
Bye,
Thanks,
guys.
guys.
Thank you.
Thank you.
Bye.
Really appreciate it.